Hello, everyone, and welcome to the Biosynapse podcast. Today, we're embarking on a deep dive into, well, an area of science that genuinely feels like the future is already here. Our focus is a really groundbreaking scientific research article. It's titled Advances in Environmental Biotechnology with CRISPR-Cas9, Bibliometric Review and Cutting-Edge Applications. Quite a mouthful, but fascinating stuff. This incredible work comes from Jessica Verdes-Odo Prado and her colleagues. And it was published in Discover Applied Sciences just this year, 2025. That's Volume 7, Issue 167, for anyone wanting to look it up. Now, what if we told you that the solution to some of our planet's biggest environmental headaches, things like climate change, food scarcity, might actually lie in rewriting the very code of life itself? It sounds like pure sci-fi, right? But it's rapidly becoming, cutting-edge reality. And here's something from the paper's abstract that really jumped out at me. Research into this specific tech, CRISPR-Cas9, has just exploded. We're talking a 30% jump since 2014. And it's not just one or two places. There are major global contributions. This isn't some niche little corner of science anymore.
It's a huge global effort. It signals this massive shift, doesn't it, towards actively trying to, use biology to fix some of the damage we've done. So our mission today is to sort of demystify this powerful tool, CRISPR-Cas9. We want to discover exactly how environmental biotechnology is using it to help hit those big UN Sustainable Development Goals, right, the SDGs. And you can think of these not just as like a list, but a whole blueprint, really. 17 interconnected goals, zero hunger, climate action, all aimed at a sustainable future by 2030. And honestly, don't worry if your last biology class was a distant memory. We promise we'll make these complex ideas not just accessible, but hopefully genuinely interesting. Okay, so to really get why tools like CRISPR are such a big deal, we probably need to set the stage first. We need to face the environmental pressures that make this tech so vital right now. Our planet is up against some really urgent challenges, and let's be honest, they're largely down to human activities. We're talking massive deforestation, and that's not just losing trees, is it? It messes up water cycles, destroys habitats.
Completely.
Yeah.
It has huge knock-on effects.
And then there's our reliance on fossil fuels. We all know that pumps greenhouse gases up, warming the planet.
Rapidly.
Plus, water pollution everywhere, oil spills, mining runoff, hitting our food and water supplies directly.
The scale is quite sobering. The FAO figures you mentioned, nearly 1 billion people facing severe food insecurity in 2022. That's staggering.
It really is.
Yeah.
And the old ways of trying to fix things, they often fall short, or sometimes even make things worse.
Exactly. And this is precisely where environmental biotechnology comes into the picture. It offers not just, different options, but genuinely sustainable ways forward. This whole field is about using life itself, microbes, plants, specific biological components like enzymes to develop new sustainable answers. Answers to our biggest environmental problems, whether that's cleaning up pollution, creating renewable energy, or managing waste better. It's about working with nature's toolkit, but giving it a boost, making it more targeted.
And inside that big biotechnological toolkit, there's one tool that really stands out. It's got this incredible precision, almost like a superpower, CRISPR-Cas9.
That's a great way to put it. Imagine having a molecular word processor for genes, something that lets you edit the genetic code of an organism with incredible accuracy. That's basically CRISPR-Cas9, the CRISPR part. Think of it as a kind of genetic GPS. It guides the Cas9 enzyme, that's the actual molecular scissor, to a very specific spot on a DNA strand.
So it knows exactly where to cut.
Precisely. It allows scientists to, edit or rewrite the genetic code like never before. It's really accurate. Fun fact, this system was actually first noticed way back in 1993 in these tiny ancient microbes called archaea.
Wow, that far back.
Yeah, but its huge potential as a genome editing tool that wasn't really clicked onto until around 2011, and its impact was so massive it nabbed the Nobel Prize in Chemistry in 2020. It truly represents a fundamental shift in how we can interact with life's code.
Okay, so understanding what it is, that's one thing. But how did it evolve into this powerhouse for environmental solutions? This paper does a great job charting that journey, doesn't it? It walks us through 3 key stages.
It does, yeah. From the early days right up to the cutting-edge stuff happening now.
So stage 1, the early days. Let's say roughly 2011 to 2015. This is when CRISPR-Cas9 first pops up as this innovative gene editing tool. And back then, the main focus was really agriculture, right? Modifying crops to resist diseases better, handle environmental stress like drought.
Making them tougher.
And one really cool application the paper highlights from this time was using CRISPR to help plants tolerate heavy metals, things like cadmium and arsenic.
Right. And this wasn't just about making the plants sort of survive in contaminated soil. It was smarter than that. This was key for something called phytoremediation. Scientists engineered the plants to actually suck up these harmful metals out of the ground.
Like tiny toxic waste cleanup crews.
Exactly. It's turning plants into living filters, pulling pollutants right out of the earth. It's a really elegant, sustainable approach to dealing with industrial contamination.
Okay, so that's the foundation. Then comes stage 2, maybe 2016 to 2020. The tech starts, as you said, stretching its legs, tackling bigger, broader problems.
Yeah, it really diversified.
So things like controlling invasive species that helps protect ecosystems, maybe reduces the need for harsh pesticides.
Precisely. And also big steps in optimizing algae and other biomass for what they call fourth-generation biofuels.
Fourth generation. Okay, what makes them different?
Well, these aren't your grandfather's biofuels. Unlike earlier types that often competed with food crops or needed lots of land, these often use genetically engineered microbes or algae. These tiny organisms are designed to be super efficient at capturing CO2 and turning it into energy. Sometimes the whole process can even be carbon negative.
Wow, okay. So they actually clean the air while making fuel.
Potentially, yes. It's a really exciting area. And during this stage, CRISPR was also used to help staple crops like rice and wheat use nitrogen fertilizer more efficiently.
Why is that important?
Well, excess nitrogen fertilizer runoff is a major pollutant, getting into rivers and oceans. Making plants use it better means we need less fertilizer in the first place.
Ah, got it. And still working on disease resistance in crops too, I assume. Protecting the food supply.
Absolutely. That remained a key focus, making our food sources more robust against pathogens.
Which brings us to now. Stage 3, 2021 to the present, contemporary times. CRISPR-Cas9 seems to be juggling multiple big challenges all at once.
It's really operating on several critical fronts simultaneously now.
Like climate resilience engineering crops that can actually handle the extremes climate change is throwing at us. High temperatures, drought, salty soils.
Exactly. Tailoring them to survive tougher conditions.
And in sustainable agriculture, it's tackling that age-old problem of pesticide overuse. I think the paper mentioned that's been an issue since, the 1940s?
Yeah, mid-20th century. So now scientists are creating plants that are naturally pest-resistant. Imagine needing far fewer chemical sprays, maybe none at all for some pests.
That would be huge.
Yeah.
And you mentioned modifying plant lignin. How does that help?
Right, lignin is a tough component in plant cell walls. Modifying it can make it easier to break down plant waste, which is useful for waste management, and it also makes producing bioethanol, another biofuel, cheaper and more efficient.
Okay, that's a powerful overview.
Yeah.
When you mentioned bioremediation earlier, cleaning up pollution, can we dive a bit deeper into how CRISPR actually does that?
Sure. So bioremediation using CRISPR often involves enhancing the natural abilities of plants or microorganisms. We tweak their genes so they get better at absorbing or breaking down pollutants. For instance, targeting specific genes like NAS1 can boost a plant's ability to take up a whole cocktail of toxic metals: cadmium, copper, iron, manganese, nickel, zinc.
So you make them hungrier for the bad stuff.
Essentially, yes. Or other genes like metallethionins help the plant tolerate higher levels of metals like cadmium, copper, and zinc once they've absorbed them. And it's not just plants. We can edit microbes too, like editing candida yeast to help it metabolize petroleum. Think about oil spills.
Wow. And wastewater. You mentioned something called Vander.
Yes, the Vander system. That's a really clever CRISPR-based tool designed specifically to find and destroy antibiotic resistance genes, or ARGs, in wastewater.
Why is that important?
Because ARGs are a huge problem. If they spread from wastewater into the environment and get picked up by bacteria that cause disease, our antibiotics might stop working. We get superbugs. It's a major global health threat and an environmental one.
I see. So CRISPR is helping fight superbugs at the source.
It's one promising approach, yes.
Yeah, and it's not just fixing problems after they happen. You mentioned it helps us spot them quicker, too. Biosensors.
Right. CRISPR-Cas-based biosensors. Think of them as super sensitive environmental detectives. They allow for really rapid, precise detection of specific contaminants. You could test water quality, soil health, assess an ecosystem almost in real time.
Like an early warning system for pollution.
Exactly. With unprecedented sensitivity and specificity, it lets you intervene much faster.
Okay, let's connect this to the really big picture.
Right.
Climate change. Methane is a huge issue, isn't it? Even more potent than CO2 in the short term.
It absolutely is. Roughly 28 times more potent over a 100-year period. And CRISPR-Cas9 is taking it on directly. Most methane comes from agriculture, livestock, rice paddies, plus energy production and landfill waste.
So how does CRISPR help there?
Several ways. Modifying microbes in crops. For example, enhancing nitrogen use efficiency in agriculture, as we mentioned, can indirectly cut methane produced by soil microbes in waterlogged oxygen-poor conditions. For rice, specific genetic edits have shown they can cut methane emissions from paddies by up to 25.5%, basically by changing how the plant grows and transports gases.
That's a significant chunk.
It is, and perhaps even more exciting is work on methanotrophic microorganisms.
Methano what, ma'am?
Methanotrophic. It just means methane-eating microbes. They naturally consume methane.
Okay, nature's methane cleanup crew.
Precisely. And scientists are using CRISPR to engineer these microbes not just to eat methane, but to turn it into valuable chemicals like mevalonate or lactic acid, turning a potent greenhouse gas into useful products.
That's incredible, turning pollution into products.
It's a really promising avenue in the circular economy concept.
So with all this amazing research happening, who's actually doing it? Where are the hotspots? The paper did that bibliometric analysis, right? Like mapping the research landscape.
Yes, exactly. It crunches the data on publications, citations, collaborations to see who's leading the charge. And the data clearly shows, perhaps unsurprisingly, that the US, China, Germany, and the UK are really at the forefront of CRISPR environmental biotech research.
Any specific institutions mentioned?
Yeah, places like the University of California System, the Chinese Academy of Sciences, major research powerhouses.
But is it just the usual suspects?
Well, that's what's interesting. While those big players are dominant, the analysis also highlights significant contributions emerging from developing countries. Places like Colombia and India are making real strides, especially in areas like making crops resilient to local environmental stresses or using plants for heavy metal cleanup.
So the technology is spreading, becoming more accessible.
It suggests a kind of democratization, yeah. Innovation isn't just confined to a few wealthy nations. Local scientists are adapting these powerful tools to tackle their own specific environmental challenges. That's a really positive sign.
Definitely. But it can't all be smooth sailing, right? This tech sounds incredibly powerful, almost like playing God sometimes. What are the hurdles, the speed bumps, when we try to actually use this stuff out in the real world?
You're absolutely right to ask that. It's definitely not without its challenges. One major hurdle is navigating the regulatory landscape. The rules for genetic engineering vary hugely from country to country, and they're often still evolving.
So what's allowed in one place might be banned in another.
Exactly. Then there's public acceptance. Genetic modification can be controversial, and winning public trust and understanding is crucial, especially when you're talking about releasing engineered organisms into the environment.
Particularly in areas with high biodiversity, I imagine. You need to be incredibly careful about unintended consequences.
Absolutely. The ecological risk assessment has to be rigorous. Ethical considerations are paramount. It's a constant balancing act, harnessing the power responsibly.
So bearing all that in mind, the huge potential and the necessary caution, what does this really mean for our future? How does it all tie back to those big UN goals?
Well, CRISPR-Cas9 really emerges as this incredibly versatile tool that can hit multiple sustainable development goals at once. Take food security, SDG 2, zero hunger. Developing climate-resilient, disease-resistant crops means more stable food supplies, less reliance on chemicals, higher yields that directly helps feed people sustainably.
Makes sense. What about clean energy?
That links to SDG 7, affordable and clean energy. Those fourth-generation biofuels we talked about using engineered algae or oil seeds offer a real pathway away from fossil fuels towards cleaner energy sources.
And environmental health generally. Cleaning up pollution, monitoring.
Right. That touches on several goals, like SDG 6, clean water and sanitation, or SDG 15, life on land. From cleaning toxic sites with phytoremediation, to early warnings with biosensors, to tackling methane, a major driver of climate change under SDG 13, climate action. It's a remarkably broad toolkit for sustainability.
And looking even further ahead, the paper mentions some really futuristic possibilities.
The future perspectives are incredibly exciting. Imagine coupling those advanced biofuels with carbon capture systems. You start moving towards a truly circular economy where CO2 isn't just waste, it's a resource.
Wow. And integrating it with AI.
Yes. Combining CRISPR-Cas9 with artificial intelligence and machine learning. Think about AI helping to pinpoint the exact tiny genetic change needed for maximum effect, or using ML to predict how an engineered organism might behave in a complex ecosystem, or analyzing sensor data instantly.
So AI helps make CRISPR even smarter and safer.
That's the idea. Making interventions more precise, monitoring more effective. But as the paper rightly stresses, the potential is huge, but so is the responsibility. We have to constantly weigh the benefits against the ethical considerations, the regulations, the potential ecological impacts. Responsible stewardship is key.
So wrapping this up, what's the big takeaway? Our deep dive into this paper really shows that CRISPR-Cas9 is, well, it's more than just another lab technique, isn't it? It feels like a genuine beacon of hope for a more sustainable future.
I think that's fair.
Its sheer versatility is astounding, helping ensure food security, fighting invasive species, pushing towards cleaner energy with advanced biofuels, and just making us way better at monitoring and cleaning up our environment.
And the paper's core conclusion really reinforces that. The authors state pretty clearly that CRISPR-Cas9 has already made significant diverse impacts in environmental biotechnology. It's driving progress towards sustainable solutions that align with those global development goals. And they particularly highlight the potential of CRISPR-based biosensors as something to watch for the future of environmental protection.
It leaves you with a pretty profound thought, doesn't it? As we get better and better at understanding and even editing the language of life with tools like CRISPR, maybe the biggest question isn't just, can we fix our environmental problems anymore? Perhaps it's more about, how will we choose to redefine our relationship with the natural world now that we actually have the power to rewrite parts of its code?
That's a powerful question to ponder.
Thank you for joining us on this incredible journey today. See you in our next deep dive.